In this session on virology and STIs, we're going to have a little look at herpes simplex virus. Now, as you know, there are two main herpes simplex viruses. There's HSV-1, which leads to cold sores, usually around the mouth region, and HSV-2, which leads it down into the reproductive system, so the external genitalia. So in this lecture, we're going to look at the pathogenesis of these HSV infections. We're going to look at the epidemiology, diagnosis, and treatment initially, before looking at the structure and replication, which involves obviously the latency period and its subsequent reactivation. So there are lots of herpes viruses. There's HSV-1 that we all know about, so that's cold sores or oral herpes. It can also lead to corneal blindness, so herpes keratitis. You have HSV-2, which leads to your genital herpes. Your variola zoster virus, which is chickenpox and shingles. And variola zoster virus is obviously, there's a vaccine against variola zoster virus or chickenpox that's used in Australia, but it's not used in all countries. So, for example, they don't use the chickenpox vaccine in the UK. And you have Epstein-Barr virus, which can lead to glandular fever. It's also known as the kissing disease, where people get fever, sore throat, and fatigue, for example, and also to Burkitt's lymphoma, which is a solid B-cell tumor in immunocompromised individuals. And that's shown in the top right-hand side there as an image of it. Interestingly, of course, variola zoster virus, it is the only airborne herpes virus, and hence why we're talking about you getting vaccinated. And following a primary infection, all herpes viruses are capable of setting up a latent state of infection in sensory ganglia neurons. Hence, they can reactivate. We're looking at how common these are. Human papillomavirus, if you're looking at numbers in the US, we're looking at about 20 million. Genital herpes, HSV-2, you're thinking about 45 million. And then you've got chlamydia and HIV, for example, as other prevalent STIs. So as you can see, herpes is by far the most prevalent STI. And in fact, it's one of the most commonly sexually transmitted STIs in the developed world. And in the US, up to 1 million people become infected each year. And again, thinking about US numbers, just because it's an interesting, it's good to know some types of numbers, and they would relate to Australian numbers in terms of demographics. It's estimated that about 1 in 5 Americans over the age of 14 are infected with genital herpes. It's also very underdiagnosed. And so when people become symptomatic, they come up with different excuses or different reasons for those symptoms. So the most common thing that women will say when they're symptomatic is that they have a yeast infection, they have vaginitis, they have a UTI, a menstrual complaint, heat rash, allergy to condoms or to sperm, and irritation through bike seats and shaving, for example. Now, some of those reasons are exactly the same in men. But they also say that the irritation to bike seats or tight jeans, for example, allergy to condoms, they'll just say, oh, it's just my normal itch. Or they'll say it's an insect or spider bite, or maybe down to hemorrhoids. So there are lots of potential things that men say they have when in fact they have genital herpes. So if we're looking at both HSV-1 and HSV-2, primary infection usually affects the mouth or pharynx or the genitals in the case of HSV-2. HSV-1 reactivation mainly affects above the waist, if we want to think about it like that, and HSV-2 below. But there is overlap. And that's because HSV-1 can cause genital herpes in about 10 to 20% of cases. So therefore, obviously, things like oral sex would be a risk. Oral sex would be a risk for that. HSV-2 is mainly through sexual transmission. In addition, the lesions initially are clear and they contain the infectious virus, and then they can become encrusted and ulcers can potentially develop. And of course, there are lots of potential reactivation factors, such as stress and fatigue, infections and immunosuppression, sexual contact, temperature changes, menstruation. So your temperature changes, things like you might burn your mouth with hot coffee or get sunburn, for example. Other causes include malnutrition, pregnancy, lactation, and as we've talked about, those physical and emotional stresses. So It's thought that in developed countries, about 20 or 25% of the adult population are infected with HSV-2. That doesn't mean that they know they have HSV-2, of course, because less than 25% of anti-HSV-2 antibody positive individuals give a clinical history of symptomatic genital herpes. So in other words, less than 25% of people who have genital herpes actually give a history of having had genital herpes. So they just don't know that they have it in most cases. But asymptomatic shedding from anti-HSV-2 positive individuals occurs even without that positive history. So in other words, you may be asymptomatic, but you can still shed HSV-2 viruses out and therefore pass it on to an individual. The virus itself, when you're asymptomatic, shedding is usually very low, so it has a low titer, and it's transient. It doesn't last very long, so up to 24 hours as a general rule. And recent evidence suggests that the transmission rate is about 10% per year to uninfected partners. And if you're looking at the primary, you remember we said that HSV-1 can also infect the genitalia as well. Well, primary genital infections with HSV-1 is indistinguishable from that with HSV-2. So you don't know which virus you're actually infected with, though obviously it is most likely to be HSV-2. In terms of diagnosis, it's also very difficult to distinguish whether it is specifically HSV-1 or HSV-2, though you can make general assumptions based on a history and physical exam. That'll help to give clues to diagnosis. So like I said, it's very hard to distinguish that primary infection from recurrent disease. And that's because if you're looking at this, you have your antibody titer, you have your detectable level. And you already know that people, when you first get an infection, you produce IgM, and that goes up, and then IgG kicks off and you get your memory forms, your memory IgG. So IgM normally in most cases shows a current infection and IgG shows a previous infection. But patients in HSV-1 and 2, the patients with a primary infection, 25% of them are detected with both IgM and IgG. And likewise, patients with a recurrent infection, again, 35% of patients are detected with both IgM and IgG. So again, that's why it makes it very hard to distinguish between a primary infection and a recurrent infection. As we said, a history and physical exam can give clues to the diagnosis, and then you can actually measure for it. So you can take vesicles, take the vesicular fluid, you can culture it, you can do PCR, electron microscopy, or immunofluorescence in an attempt to diagnose the herpes virus type. So how is it treated? Well, treatment is used, treatment is acyclovir as a general rule, and it's the most common treatment, and it arrests DNA elongation. Now, herpes simplex virus, it replicates within the nucleus or the nuclear plasm of cells. Okay, so this is where it replicates. So when HSV gets in, this is where it goes to make its DNA and to make its messenger RNA. So this is where it replicates. In particular, this is where it elongates its DNA, and it uses its own DNA polymerase. Okay, so it doesn't use the the host DNA polymerase. It uses its own DNA polymerase. So how does acyclovir actually work? Well, acyclovir is basically a nucleoside analog, so it mimics GDP or deoxyguanosine triphosphate. So that's what it's mimicking. And it selectively inhibits the replication of HSV types 1 and 2 and variola zoster virus as well. So after the initial intake into the cell, it's converted into a acyclovir monophosphate. And this is done via a virally encoded thymidine kinase. So basically this drug that we're giving to people or you're giving to yourself is being converted into a phosphorylated form by a viral kinase, so thymidine kinase. This step doesn't occur to any significant degree in uninfected cells, and therefore it lends specificity to the drug's activity. Because remember, your regular uninfected cells don't have this viral thymidine kinase, so therefore you're not getting that process occurring in uninfected cells. So we've now got an acyclovir monophosphate, and this is then converted into a diphosphate and then a triphosphate by cellular kinases. So kinases are just enzymes that add phosphates on. Phosphatases are enzymes that take phosphates off. So basically, it becomes a cyclovir triphosphate. So this acyclovir triphosphate then acts as an analog to the D-guanosine triphosphate. So that's like GTP. And that competitively inhibits the viral DNA polymerase from working. So you're stopping that viral DNA polymerase from working. So incorporation of the acyclovir triphosphate into the DNA results in chain termination. And so therefore the DNA polymerase can no longer work. Acyclovir triphosphate as well has a much higher affinity for viral DNA polymerase than for the cellular homolog, therefore again yielding a high therapeutic ratio, which makes inhibition of the native, the host DNA polymerase clinically pretty unimportant. So it's very specific to inhibiting DNA elongation using the DNA polymerase that is made by the virus. Just other little interesting fact, HSV proteins, they contain a lot more arginine than lysine, so those are amino acids. And so therefore it's thought that a higher lysine-arginine ratio within your food helps to suppress the life cycle. So that's the first part. The next part is we're going to look at the structure and replication and also latency and reactivation. So if we're looking at the structure of viruses, you remember that a virus has got a nucleoprotein core. And in this case, it's a double-stranded DNA virus, so it has double-stranded DNA within its nucleoplasm, within the nuclear core rather. Surrounding this, it has a capsid and it also has an envelope. Now, in between the envelope and the capsid is a tegument, and this tegument contains lots of proteins, some of which will remain in the cytoplasm. For example, some of those tegument proteins will shut off host protein synthesis, so you can make even more viral proteins because you're not making any of the host proteins. And basically, the tegument is really just a cluster of proteins that line the space between the nucleocapsid and the envelope itself, and it's present in many envelope viruses. Looking at the envelope, there are lots and lots of membrane proteins that this virus expresses, and about 5 of these are involved in HSV entry into the cell. So it requires lots of those proteins to work to get in there. And the virus makes about 50 proteins altogether. The other interesting thing, of course, humans are the only natural host for herpes viruses. So what's happening at its replication? We're going to keep this very, very simple. So the virus enters through membrane fusion. So in other words, the plasma membrane, the cell membrane, fuses with the membrane of the envelope of the virus. And that's mediated by a variety of viral proteins and host receptors. This then releases a partially uncoated genome into the cytoplasm of the cell. And then once it's within the cytoplasm of the cell, that viral double-stranded DNA is incorporated through nucleopores into the nucleoplasm. So remember, this is where replication takes place and where the DNA polymerase, the viral DNA polymerase is. You then get gene transcription and you get DNA replication. So you're getting transcription, which is making viral messenger RNA, which obviously then has to go back out to the cytoplasm where it interacts with ribosomes and it makes the viral proteins. And you also replicate the DNA in there. Now, you can then enter your productive lytic cycle. So that basically just means that the DNA and the viral proteins come together, they assemble, you release the virus and you make the virus and it's released. But of course, over time, those cells will die. And what the virus does, it actually goes back, it sets up a latent infection within your sensory neuron ganglia. And so that's what we're going to talk about in a second. And the virus divides very slowly division does occur in these cells, but it occurs very, very slowly. And it's stimulated by the presence of a variety of enzymes that increase the nucleotide pool. So that's your A, G, T, and C, obviously. So what's happening? So division in the slowly dividing cells, the herpes virus itself contains the enzyme required for increasing that pool of nucleotides. That's what I just mentioned. Some of these enzymes are things like thymidine kinase and ribonucleotide reductase. Doesn't matter what these do. Okay, just be aware that it has enzymes for increasing the pool of nucleotides. And so, of course, these are targets for antivirals, because if you can target these enzymes, there are less nucleotides, so the virus can't replicate as easily. Infected cells producing herpes virus, obviously they don't survive. So after a productive primary infection, these herpes viruses enter the sensory neurons. And that's where they can become latent. So about 50% of the HSV-1 and HSV-2 genes are required for establishing latency, so that they can sit very quietly within those nerve cells, and also for immune evasion. And so immune evasion, it has molecules involved in that act as virokines and viroreceptors. But what are virokines and viroreceptors? Well, your virokines are proteins that basically mimic cytokines. They look like cytokines or similar extracellular immune regulators. They bind to the cytokine receptors and therefore block the action. They're basically mirroring cytokines, block the cytokine receptors, and therefore you don't get that signaling and stimulation of the immune response. You also have the viroreceptors. These are soluble viral receptors that bind to cytokines and prevent their action. So in other words, here's another one. Here is a soluble viral receptor, and it goes on to bind cytokines that are produced following an infection and takes them out of the equation, so they can no longer bind to the cytokine receptor and elicit a response. Most of these virokines and viroreceptors have been found in the large DNA viruses, such as the herpes viruses, and that's because they express so many proteins. They have the ability to express, they have more proteins than just those involved in replication, division, and assembly. So you can go through a true latency period. And so this is where the virus remains completely latent following a primary infection. It may be present in immunoprivileged sites, so the neural ganglia. So those are sites where there isn't a strong immune system present. Its genome can potentially be integrated into the cellular genome, or it can exist as episomes. And episomes are just circular DNA molecules that are independent of the host genome. So if you're looking at these, this latent period, you don't get any replication. So you can have a latent virus, or you can have reactivating virus, such as herpes and shingles. So if you're looking at it, you get these latent periods where it's quiet, then you get reactivation, latency, reactivation, latency, reactivation. So where are those latency sites? Well, HSV sets up a latent infection in the sacral ganglia, and you get asymptomatic viral shedding can occur independent of outbreaks, and we talked about that a little bit earlier. In HSV-1 can set up latent infections in the trigeminal ganglia, and shedding occurs for about 24 hours during an outbreak. And again, this is most commonly, as we've talked about, is on the lips, is where you get the active lesion forming. So how does this happen? Well, HSV travels along the sensory neurons, innervating the primary site of infection to the nucleus, where they remain dormant. So here are your epithelial cells, which are undergoing their lytic cycle. The virus is being released, and the virus then enters into these sensory neurons. You get retrograde movement of the capsid along the axon, where it moves into the nucleus, where it goes to the nucleus, where it can remain dormant. And in HSV, the genome persists as an episome, so that's independent of the DNA, double-stranded DNA. So here it is, you've got your latent HSV episome, and here you've got your sensory neuron DNA, so that's your cell DNA. And it also makes a couple of little proteins, and these are called LATs, or latency-associated transcripts. And these are there, they're thought that they may be involved in preventing apoptosis of the infected cells, but there's no viral replication going on at this point. And following reactivation, and there are lots of things that can stimulate that, so heat, for example, is something we talked about, stress is another one, the virus is synthesized and travels along the axon to the skin surface. So you get that appropriate stimulus, it reactivates the virus from its latent state, and you get anterograde transport, moves the capsid back along the capsid, where it can go on and infect your epithelial cells. So it initiates a general infection at the peripheral site, where limited virus replication takes place until the host defense systems respond and suppress it, or until you give someone a cyclovir, of course. It's not known whether the reactivation event involves killing of a few neurons or not, but most evidence does suggest that the frequency and reactivation decreases with time, suggesting that there may be some death of neurons.